Thank you, Blandine. Good morning, everyone. Okay, so we are going to talk about a comparison, as Blandine said, between non-target and broad-spectrum target NGS for advantageous virus detection. These are two strategies that we have at the Polar Lake Institute, and we have compared both of them. So first, the typical disclaimer. So everything that I'm going to say here is not the position of the Polar Lake or any regulatory agency. It's practically my experience and my own thoughts. So the introduction, first, what was the problem and why we started that at Polar Lake is not only for advantageous virus detection in biological products related to vaccines and so on, but also in blood-related or blood-derived products. And the main issue that we encounter is that the plasma pools that are made to obtain all these derivatives from blood products are a mixture of a lot of donors. Could be from 3,000 to 36,000 donors in one big tank. And from there, we need to search that they are free of viruses. So one of our highest challenge was to have a very deep sensitivity. And of course, the other part they use in vaccines and biomedicines that yesterday and the day before it had been addressed a lot. So I will not go further in detailing here. So first, sad true that all of us know, but we don't like to talk about it often, is that the unbiased sequence or this agnostic doesn't exist. Every single step that we put in our workflow, it's going to bias for one type or other type of viruses. I just put, sorry, a reference here. This is a paper, very nice, where they compare different ways of making a pre-amplification step, trying to increase the viral part to increase the sensitivity of the detection. And as you can see, depending on which one they use, there are different virus enriched, either the single-strand RNA, the double-strand RNA, the DNA, and so on. So really, really unbiased, and this is just an example in this pre-amplification, but also in destruction, in the library, in every single step, we introduce bias, even in the bioinformatics. So before that, I want to just explain yesterday, when we heard from Malé and Winel about the pharmacopoeia chapter, we have a dedicated part of targeted, and this is divided in two parts, in analysis, but also in the wet-pad lab. In this part of the laboratory, normally we think in target NGS as a short spectrum or very selected viruses that we can target either by amplifying them with specific primers or by capture them also with specific primers attached to a tag. What we have in the Institute, and this is due to a collaboration with Columbia University, is a system called BIRCAP-SecVert that is a broad spectrum capture system. So we don't select a very few viruses. To develop this system, it was selected more than 207 taxa from all the viruses that were known or predicted that could infect any vertebrate. These were then only selected for the coding sequence and group these taxa by homology from more than 85 to 96%. And after the grouping, they will make the probes that have a length around 70 and 79 nucleotides in average, but it could be from 50 to 120 nucleotides. This BIRCAP-SecVert has two versions, so the newest version have around 700,000 hybridization probes. And how is it made? This is not, of course, the probes, but it's just to give you an example. You can imagine that these are different viruses that have the same homology. Then they don't make really a consensus. They just search, okay, these have the homology here, and these will be the probes design. The probes are not one probe per virus, taxa, or per group, or per homology. There are many, many probes for each of these groups, because if each of them has, as I said, an average 79 nucleotides, and then in the sequence, there is like a hole, let's say, that we don't have a probe from 25 to 50 nucleotides, and then comes the next probe, and the next probe, and the next probe. What allows that to also catch new viruses that has certain level of homology. Yesterday, I had actually a meeting with a group in Colombia, and we were discussing about this database that was presented by PathoQuest, about these viruses that was created artificially in silico by evolutionary patterns. And they made some analysis, just quick analysis yesterday in the evening, and they found that to really have something that it's biological plausible, it cannot be more than, it cannot have less than 35% homology. And in the silico experiments that we have done with these probes, we are able to catch all of these viruses that have at least 35% homology. So that will allow us also to capture new viruses. And this is published, Dr. Ian Lipkin, whose group has developed this technique, have many papers of the discovery of around 2,000 new viruses with this technique. So how is our laboratory workflow? First, the non-target NGS. This is the typical workflow that everyone's know. I have to say that we have modified a little bit. So for the extraction part, we don't extract separately RNA and DNA. We make just one total nucleic acid extraction. But then in the pre-treatment, they are separated, and part of it is going for the cDNA synthesis and second strand, and the other part is just going to be treated as was only DNA and goes for the second strand, and then I mix it to create the double strand. And then for the non-targeted, we have a pre-amplification step, and then goes to the fragmentation and repair, ligation, and all the parts that you already know until the sequence and the bioinformatic analysis. What are the difference with our target approach? For our target approach, and this is something that we learned from the non-target approach, as I said, every step can make a bias. And we found that if we hybridize, not hybridize, sorry, if we denaturize the RNA and DNA only, or the RNA mainly, at 75 degrees, that was not enough to have a good sensitivity for orthoreovirus. So we increase the temperature, that works really nice, but that could diminish how we are able to capture the other viruses. That's why we have the total nucleic acid that I said, but we divide the sample in two, actually in three. Two of them are treated as were RNA with two different times of this here, two different temperatures for the nature, then are mixed, do the cDNA, and then are mixed with the third part to make the second strand DNA, and then we have our sample together to start the library. The library goes like every normal library. We don't have any difference in that part. And after the library is made, of course, you know that in the last step of the library, we make a PCR, but we make here a PCR with few cycles, 11 or 10 cycles, no more, because up to here, because we don't do any pre-treatment, I forgot to say in the other workflow, in the traditional one, in the non-target, we make a pre-treatment with benzonase, nuclease, and so on to try to reduce the host. Here, we don't need to do that because that's going to be in the capture part. So we do the library with everything there, with all the background and so on. That allow us to don't have any problem for the excess of adapters and so on, because we have enough material there. And at the end, we make this low cycles PCR, because if the ratio from virus to human, in our case, is, for example, 10:1, then if we do a very extensive amplification, this ratio is going to be a very bad bias for the virus. That's why we keep it low. But then we make the capture. The capture is basically an hybridization that we make with the probes that are marked with biotin. And then we capture with the streptavidin bits. We wash it and so on. And after the capture, we make a second amplification through the adapters as the normal amplification that is done in the library. But here we increase to 16 cycles because we have mainly viruses and not other things. And then goes for the sequencing and for the analysis. So one of the first challenges that we encounter with this is the sensitivity. This was normally used in the clinical field for detection of viruses, of different pathogens in human samples. So they normally use 48 plex. So they multiplex with 48 samples. We were afraid that was too much for the sensitivity that we wanted. And we make a test, making a duplex, 12 plex, and 24 plex. At that time, we were not having yet the WHO standards. So we just made with some pools and with the same samples, we knew what was there after sequencing, and we compare the total hit length, the coverage, and the line rates. And as you can see, if we do the 24, it's enough to detect all of them. And to do it in 12 and in 2, it doesn't tell us more information, so we don't detect more viruses. We just have a better coverage in some of them, but very few. The difference is not enough to say, okay, it's better to do a duplex or 12 plex. That's why we stay with the 24 plex. We did, of course, the 48, but it was too low. That's why we didn't put it here. So our technique stay with 24 plex for these plasma pools. And now the comparison. For the comparison, we have used the WHO standards, spiking in plasma from blood donors in plasma pools, because that was our main goal. So from the first virus, the feline leukemia, you will see in all the viruses the 5 replicates, the comparison with target and non-target, and the different viral loads from 10 to the 1 genome equivalents per milliliter to 10 to the 6. If it's green, is we detected. If it's red, we were not able to detect it. The LOD we determine as at least 3 detections as the lowest one from the 5. And here you can see the coverage of the full genome. The blue dot is the target, and the green square is the non-target. And you can see also for the different viral loads, how much or which percentage of the full genome coverage we were able to have in the 5 replicates. And from the map reads, from million reads, also to have a proper comparison, because in the in the non-target approach, we use 100 million reads per sample. As in the target, because we make a multiplex of 24, we use only an average of 17 million. So as you can see, the sensitivity here was up to 10 to the 3 for the target and up to 10 to the 4 for the non-target. For the human coronavirus, OC43 was up to 10 to the 1 for the target and up to 10 to the 1 as well for the non-target. But here the coverage was not so good. The number of reads was good enough, so we can determine it was positive, but the coverage was not ideal. I can tell you why later on. And for RSV was the same case of OC43, so the sensitivity was a little bit not so good, was 10 to the 3 for the target, and also for the non-target. The coverage was also a little bit problematic. By Epstein-Barr was really good, 10 to the 1 for the target and 10 to the 3 for the non-target, with very good coverage, up to 10 to the 4. And in the comparison of the map reads per million reads as well. Minute virus of mice, we have up to 10 to the 2 to the target and 10 to the 3 for the non-target. For the porcine circuit, 10 to the 1 for the non-target, for the target, sorry, and 10 to the 3 for the non-target. No, yeah, 10 to the 3. And the mammalian orthorheo, 10 to the 1 for the target after we increase the temperature. Before it was 10 to the 3. And for the non-target, 10 to the 4. And as you can see, we can have also even in the 10 to the 1, very good coverage in some of the replicates. Then we wanted to test the breadth of detection. Of course, the 7 virus panel is intended to demonstrate that you have the breadth of detection, but because we have a target approach, we wanted to challenge a little bit more. So we use a 21 virus panel in-house, and because we use plasma pool, we have human pagivirus in mainly all of the plasmas. That's why we put it in yellow, because it was not intentionally put it in, but we found it as well. And some of these viruses are contaminated with parainfluenza 5 that we also found. So for the target and the not target, for both, we were able to detect all of them. So the breadth of detection is not reduced, at least for this 21 virus panel, in our target approach. Then the workflow summary. So if we use the target approach, we don't use a nuclease sample pre-treatment. If we use the standard metagenomic or non-target, we use it. The illusion was exactly the same. The pre-amplification step, as I said, it was modified from the beginning, so now we don't use it also for the standard metagenomics. The denature temperature, we have multiple, but here we have only one. The retrotranscription step, we extended a little bit in our target approach. Both are made the second strand by Nick translation based. The library is a different kit, but works exactly the same. PCR amplification, we use 11 cycles, and for the non-target, it's variable. It's according to the measure that they made, how many cycles that it will give. Of course, in the target, we have hybridization capture. In the not-target, we don't have it. There is a second PCR amplification in the BIRCAP-SecBERT, but here it's not. Both of them are sequencing the Illumina. Now we are sequencing the P3. Before we were sequenced in the P2, these ones, and in the P3, these ones. And as I said before, we expect here 70 million reads per sample, and here 1 million reads. If we go for the cost of it, mainly are the same cost, unless the difference is, of course, the hybridization that we don't apply here, but this cost is overtook by the cost of the sequencing itself. So if we use a pre-treat flow cell, sorry, in the last version, it costs 100 euros per sample. Now in the new chemistry, it costs 76, while in the standard metagenomic, because we only multiplex 10 samples here, in the past chemistry is 600, while in the actual chemistry is 457. So these are the final cost with the old and the new chemistry. And this is the savings. So if we use in the capture system, we're saving in the old chemistry 55%, and in the new chemistry 48% of the cost. And I know that some of you even sequence at 600 or 400 million reads per sample. So if you sequence like that, practically you are using a flow cell entirely for just one sample. So that will be, if you use the capture system, at 98.2% of reduction cost. Then we wanted also to challenge with real samples, and this is an ongoing project. So yesterday was an update of the data. I was analyzing, that's why I sent my presentation midnight. Sorry for that. So this is just a brief summary of this new with the real sample test. So as some of you know, I'm Mexican, so we have good contacts there. And it was easy to have different blood banks to collaborate with us in different regions of the country. What is the interesting of this and why we selected like that? Because we have different climates in Mexico, and that will key for different virus findings. So we have from here, there is Durango, Mexico City, Puebla, Chiapas. I forgot one here in Cancun. And it was collected for 1 year to have the different seasonalities. Also, we collected not only blood donors, but also the ones that were rejected by any reasons, even the interview or any positive test. We pool them as 100 samples per pool, and we make a meta pools as well of 10 pools of 1,000, just to compare the sensitivity. At the end, we finished with 898 pools of 100, so 89,500 donors that we are going to analyze around the year in different seasons and in different regions. And this is just to show you how it comes, our results. This is how I saw the results when it comes from the bioinformatics. In the latest session, a colleague of mine, Martin Makine, is going to explain how our bioinformatics works, but this is how we have it at the end. So we have a list report with the taxon ID, the name of the virus according to the taxon, and the read number that we have in that taxon. To have a little bit more strict, let's say, analysis, we have a reclassification. What is the reclassification? We transform the sequence into protein or amino acid sequence by the 6 read frames, and we see if that's still passed or not for this virus. And then an additional filtering is it composed of 2 things. One is it has to have at least 3 non-contiguous regions around the across the genome to call it as passed. And also it depends on the length of the virus, but it has to have a minimum percentage of coverage nucleotides to pass this filter. The average, let's say, is 1,000, but of course for Epstein-Barr or herpes virus, it's bigger. And for viruses like parvo, that is too short, are less reads. If it fails, we consider it for the after analysis that we do afterwards, but we mainly focus on this. So then we have the coverage plots, of course. For example, in one of the samples from the donors, we found dengue virus type 1. That is a virus that is normally not tested by blood banks. And as you can see, we have here the coverage, how many reads we have, the percentage, and the depth of the coverage, and so on. This is another virus that I will say was not a true hit. It's just 32 base pairs. And GBVC, that is the PEGI virus that is practically in all our samples. After that, as you saw, here we have a button that is copy the consensus sequence. So we create a consensus sequence, and we go back to blast the consensus sequence to see if it really goes for the virus that we are looking. And for example, this was the dengue type 1. And you can see how our consensus sequence is distributed across the whole genome. So this is a true hit. This is a dengue type 1 from other pool. This was not so good as the previous, but enough to call it as a positive hit. And we found, and you will see in my last slide, a lot of human herpes virus 6 in our donor samples. And normally in our pipeline, it comes 3 times. It comes human herpes virus 6, human herpes virus 6A, and human herpes virus 6B. So how we distinguish, this was the same sample I'm going to show you. This was the reads that were aligned to the beta herpes virus 6. As you can see, it's clearly positive hit. And then in the same sample, it was the ones aligned to the accession number of the human herpes virus 6B. Goes really good. So we can say could be a 6B, but let's see if it's a 6A. These are the ones that are aligned to the 6A. So this clearly is for homology, and these reads should be aligned here. But this is how we can determine, okay, is one or the other. So just a brief summary, and as I said, it's not from all the pools. I'll just take 40 randomly just today to analyze. Some from Mexico City, others from Puebla, Chiapas, that is in the south and is practically the jungle. In Cancun, that probably many of you know is a vacation resort. So we found from the 40 pools, it's 400 donors. The 92% of the pools, so the 9% of the donors have PEGI virus. TTV, that is also a commensal virus, 97% of the pools have it, 9.75% of the donors. Herpes virus 6B, 22% of the pools, 2% of the donors. The genotype A, just 2% of the pools, 0.25% of the donors. Papilloma just in 1 in Mexico City, so 2.5%. Epstein-Barr in 2 of them, so 5% of the donors, 0.5% of the donors, 5% of the pools. Hepatitis E, that also is not tested in Mexico blood banks. We have found it in 2. We don't know exactly why, because there was in pools and we are not able to track back the donors, but these 2 pools were rejected. So are from donors that were rejected. Of course, they don't test for hepatitis E, but for any other reason, they rejected, probably by the interview or something. So we have 2%. One curiosity, we have adenovirus 11 in one of the pools with a very good coverage. And this is 2.5%. parvovirus-19 from these 40, only in 2 of them. Kaposi sarcoma, only in 1 of them. Dengue virus 1 in 2 of them from this region that I told you that is basically jungle. 1 of them in Mexico City. This is not normal, but this year I know that was an outbreak in Mexico due to the global warming. And dengue 3 in Puebla, that some regions of this state can also have a tropical climate. So with that, that is a summary, and I just want to thank the project leaders, so Johannes Blumel and Renate König, the non-target NGS that was performed by Chava, Dora, and Pauline, the target NGS basically performed by Janice, the bioinformatic group, Marcus Brown and Martin McKenna, that you will hear from here later, and the Virkapsekberg group in the Columbia University, Thomas Breeze, Kenneth Wikisa, Vishal Kapoor, and Alexander Petrosov, and of course, Ian Lipkin, that is the boss of this laboratory. Thank you very much.
Thank you, Blandine. So I will start, and as you say, we will make the distinction between what we use for release testing and investigation. So I will start to present or represent the context of Sanofi vaccine HTS advirus detection and our choice regarding in vivo substitution. And then I will leave the floor to Song for the overview of the test, the control and the validity criteria that we use, the method development and the metrics assessment, the generic validation, as well as the evaluation of new metrics that we are performing, and the follow-up investigation tool that we are using before concluding and going for coffee. So I'm sorry, maybe it will be like a déjà vu slide because I presented quite a lot and I apologize for that, but this is an overview of the traditional testing package for adventitious virus. So it's made of non-specific test method and specific test method. Each method is targeting a specific viral characteristic. So none of this method is able to detect all viruses at once. So within the non-specific method, and Johannes made a really nice overview of this with all the details regarding those methods. We have the test in animals, so the so-called in vivo test, and the test in cell culture, the in vitro test, on which we had the test for retroviruses and electron microscopy. And because this is not able to cover everything, then you have the specific test method for porcine and bovine viruses with the 9CFR, some antibody production test, and you can complement with some PCR based on your viral risk assessment. But as we all know that there is some strong limitation within those testing package. So within the in vivo test, it's only when you have viruses that replicate within this model animal and at the same time that create clinical signs that you will be able to detect those potential contaminants, meaning that you will miss a lot of viruses with the in vivo test model. Within the in vitro system, the readout are cytopathic effect or M absorption. So you need to have the right indicator cell to be able to detect this CPE, and all viruses are not making CPE or are not M absorbing. So this is a theory, and that was nicely illustrated through an NIH study that was conducted and led by Rebecca Schitt. So I saw her yesterday. I don't know if she's around, but at least we can, yes. So we are using this study for a while, and I'm happy that she's around. They did this systematic evaluation of both in vivo and in vitro against 16 model viruses. And as you can see on this figure, the longer the bar, the best detection it is. So the in vitro system is not that bad at detecting viruses, although sometimes you need to have some specific indicator cell. But the in vivo model is quite bad. It's only when the virus to detect fit its animal model that you will be able to detect it. So that's the problem with the non-specific test. And then you have to complement with your specific test method. So you have to perform your viral risk assessment to really define what your non-specific method may miss. And when you develop those specific method, of course, you have to consider viral variant, as Gibran mentioned this morning. So the conventional test package could miss a viral contaminant if you don't perform your viral risk assessment with a good accuracy. And that was really highlighted with the discovery of porcine circovirus within rotavirus vaccine, work done by an academic lab in 2010. And within this work, they highlighted that high-throughput sequencing has the potential to identify both known and unknown advanced virus by sequencing all the nucleic acid within a sample without needing any prior knowledge of the contaminant agent and this, whatever the virus. And FDA invited all licensed vaccine manufacturers to take action and consider improving their testing method if there was the need. So although our interest for HTS was prior this event, definitively this event helped us to strongly build our pipeline. Our goal was to substitute the in vivo test because we knew it was a troublemaker and it was not that sensitive. And we wanted to strengthen our viral testing package. So the idea was to substitute the in vivo and at the same time, then we can replace all the specific test method. Our choice by doing the in vivo test substitution was really focused on cell bank and viral seed testing. So the in vivo tests are performed at those stages, and so the matrix that we had to deal with were cell and harvest with more or less cell component. Our goal was to have one approach that will fit all type of matrices. So we put here back the beginning of the figure of the 2641 chapter to show you where we are in terms of the technique we wanted to approach. But since our new vaccine are produced in animal-free material, so we choose a pipeline that is able to detect any viral nucleic acid and preferentially those that are encapsulated and potentially the ones that are not encapsulated as well. To conduct the substitution, as Johannes mentioned yesterday, it's comparing apple and pear. But thanks to the 5214 chapter, there is a way to assess the two methods, the one that will substitute the other. And this substitution is made through the comparison of the specificity, the breadth of detection, and the sensitivity of the new and the existing method using a panel of well-characterized model viruses. And again, thanks to the work conducted by NIH and Rebecca, we have these 16 model viruses that were really nicely characterized and assessed within the in vivo and in vitro system. So they represent potential contaminant that could be introduced within vaccine production and a variety of viral structure, RNA and DNA genome, and envelope or non-envelope virus. We use the same protocol as NIH to both produce and characterize those stock. And by generating those material, we were able to link our HTS data with the in vivo published data. And this as proposed by the 5214 chapter. This has been published quite a while ago, so in 2020, but this is a comparison of our HTS data versus the in vivo. And so using the viral stock as representative spiking material in our detection pipeline, we were able to document the breadth of detection and sensitivity of our HTS pipeline. And our data package supports the substitution of the in vivo test by HTS. And so we were able to drastically streamline our advanced testing package and at the same time strengthen our product viral safety. And with this, I give the floor to Song to enter you into the detail of our test.
Thank you. So this is an overview of our test pipeline. This is the typical viromics/genomics approach. As Corinne mentioned, that our pipeline favors detecting the encapsulated viral nucleic acids, but also detect the non-encapsulated viral nucleic acids to a certain extent. Actually, we have been working on different types of matrices. I checked the data set. There are always a substantial fraction of the rays actually host nucleic acids because you even use nuclease, you will not remove all the host nucleic acids. As a matter of fact, if you remove all the host nucleic acids, it's challenging to get a sequenceable library because that won't meet our validity criteria for a good sequencing library. And also because we don't want to, there's a step of limited PCR amplification during the library preparation. You don't want to have too many cycles which will introduce bias. So you want to keep it as low as possible. And one more thing I want to emphasize for this pipeline is we have two internal controls, the pseudoviral particles for control for DNA virus and RNA virus. So for every single test, we add these two internal controls at the very beginning to the sample matrix before any pre-extraction treatment. So these two system suitability controls can be used to assess the performance of every single step of this test, including the pre-treatment, the nucleic acid extraction, the first, the second strand, the staging synthesis, the library preparation, sequencing, and also the bioinformatics analysis. In the bioinformatics report, we have the validity criteria for the recovery of these two internal controls are required to meet certain threshold that predefined, and it's matrix specific. So if the internal control meet the criteria and no actionable signals, then it's conform. But if there's actionable signals, then we go to the follow-up investigation, which I'm gonna talk about later in my presentation. So this slide summarizes the controls and the validity criteria for our test, and some are general to all the matrices, some are matrix specific. For general ones, the measures are implemented to avoid cross-contamination, such as the lab rooms are segregated with the dedicated biosafety cabinets for nucleic acid extraction and library preparation with unidirectional workflow. The libraries are dual indexed to avoid potential contamination from different libraries. I think that's a common practice. And so the input DNA will be quantified for the sequencing library preparation. And the validity criteria are implemented for the size and the concentration of the sequencing library during the assessment of the sequencing library step. And for the sequencing run, there is validity criteria implemented for the read quality as the minimum percentage of bases with Q score equal or greater than 30 for the sequencing run. And some controls and criteria for matrix specific includes for sequencing, there's a criteria for the total output per sample as a number of reads. So we typical sequencing P2 flow cell per sample. And for the internal controls, so we have matrix specific validity criteria for recovery of the internal controls as number of reads detected for these two pseudoviral particles. We need spiking materials for method development and validation. Here is our choice for the well-characterized viral stocks. The chosen reference viruses must represent the viral diversity in terms of structure, like enveloped, non-enveloped, nucleic acid type, RNA genome, DNA genome, single-stranded, double-stranded, circular, linear, segmented, and different genome sizes, virus morphology, and the chemical resistance. And this reference model viral stocks are well characterized for genome copy number, infectious titer, viral genome sequence, including variants, and any additional expected background signals. So it has been mentioned several times in previous talks in the last 2 days, and the panel of WHO reference viral stocks, which recommended use for the adventitious viral detection by HTS. And before this WHO panel available, we use an equivalent panel of 16 NIH viruses, which is used in the Humboldt et al. 2014 paper to compare the sensitivity of in vivo and in vitro assays. Okay, so for new matrix, we this is before validation. We do method development and matrix assessment using the representative matrix samples to evaluate the optional pre-extraction steps, the potential procedure modification for method improvement, to determine the target LOD and the specificity using the spiking materials, either 16 model viruses or WHO panel, also to demonstrate the assay robustness during the development, to define the validity criteria for the internal controls. That's quite important. We need multiple samples to understand the different levels of recovery. The assay is not quantitative. Some may argue that it's semi-quantitative, but we do try to get enough individual samples to understand some statistics, different levels of variations. Also, during the development, we can also identify the false positive generative signals potentially coming from the matrix or the testing reagents. So we can deal with this before the validation and testing. And based on the method development, some considerations for the validation, to improve the method, such as at a new step to improve the sensitivity, replace the reagent kit, or upgrade the bioinformatics software, and also provide the matrix-specific instructions for validation. For wet lab, use nuclease treatment or not. Some matrix you need lyse the cell. For the dry lab, which host to pre-screen for the bioinformatic analysis, and also very important, the validity criteria for the two internal controls. At what level or threshold you need to recover the two internal controls for assay. And then we go to validation. Need to write up a validation plan, approve a validation plan with a clearly defined validation characteristics for the LOD and the specificity that's required by the guideline, and the details of the study design, like the description of the method, materials, equipment, how you prepare the spiking pools, viral pool, and how those spiking samples prepared, those details in clearly defined in the validation plan, and also define the validity and acceptance criteria for the validation. Then you execute this plan, exactly follow the plan, the approved plan, and to demonstrate the LOD and the specificity. Typically, we have 2 or 3 spike samples to demonstrate the LOD, and in addition, 1 on-spike sample. Together with the 2 spike samples, you demonstrate the specificity. This example of a generic validation using a viral crew harvest matrix. The test is impurity test as a limit test. So the validation parameters or characteristics that you need to demonstrate is a specificity and the sensitivity, the LOD limit of detection with the spike samples. And in this case, the 16 NIH viruses were spiked at 10 to 14 copies per mil into 1 mil of the viral harvest seed matrix. And also the 2 internal controls also spiked in the matrix to control for the performance of the test. So for the 2 spike samples, we have were processed by 2 different analysts over 2 different days to capture the variation, and plus 1 negative control sample for the specificity. And this is the framework that we use to for any new matrix coming. So, do we do validation? Do we do verification? This is the framework that we define. If there's a new matrix, we first ask if an equivalent matrix has been previously validated or not. If the answer is no, then we go to the method development and the generic validation as described in the previous slides. If the answer is yes, then we ask the next question, if there's a potential impact on the LOD or specificity for the new matrix, we look at the attributes like antigen expressed, compare the manufacturing process, also the freezing medium. So anything that can impact your assay for the LOD. And if the answer is yes, there looks like will be an impact, then we go back to the development to understand the LOD and do the validation. If the answer is no, then we have at least one round to verify the LOD and also to confirm the validity criteria for the two internal controls to make sure it is expected. After that verification, we do the testing using the validity criteria defined for the equivalent matrix. So, as I mentioned in the previous slide, if there's an action or signal detected, you go to follow-up investigation. So this table, I think, is taken from the draft of the European Pharmacopoeia, chapter 2641. And for the follow-up investigation, there's 3 key questions that I want to ask. Are the nucleic acids that were found associated with the full-length intact genome? The second one is, are the nucleic acids particle associated, and are the particles infectious in cell cultures? As highlighted here, there's different types of molecular methods like PCR and other types of HTS technologies that can be used to address these 3 key questions. So the follow-up investigation is to be built based on the identified signal. So when you identify a signal, you know the sequence, then you can design your follow-up assay with dedicated tools to conclude on the replicative status of this potential viral contaminants. So the next few slides, I'm going to briefly go through the follow-up investigation tools that we are currently developing or evaluating. PCR, the first two, the sequence, it can be specific, and you can design PCR primers, oligos, understand the sensitivity as well. So we're developing a generic PCR method as an orthogonal method. So you have an orthogonal method that you can use completely different types of reagents that, to further confirm the signal, to say you want to first confirm if the signal is really in your matrix, not in the testing reagents. Leveraging the sequence data to design the primer and probes, and the sensitivity of the PCR can be estimated using synthetic templates. So then the PCR is comparable or even more sensitive than your HTS test to make sure you can find the signal. And also the multiple sample can be tested in parallel with relatively short turnaround time. So that will be very useful for your investigation if you want to identify the source of these potential contaminants. Oxford nanopore sequencing. We are also currently evaluating the ONT to assess the genome integrity. So ONT is capable of generating very long reads. We all know that. It can sequence native DNA or RNA without the need for amplification. The data analysis can be real time. But there's potential considerations that the high yield of input DNA or RNA are usually required for good sequencing run with good output and good quality. And we are also evaluating a single-stranded RNA-seq method, which is very useful to detect. It can detect the viral RNA with directionality And so if you detect signal, you know what type of virus it is, and then you can determine the viral replication based on the type of the viral genomes. Positive cis, negative cis, single strand, double stranded, DNA virus, then you look for transcripts. But the potential consideration is that transcript may be degraded in some matrices. Is also matrix specific. And also typically the high yield of input RNA is required to generate a good sequencing library. Okay, with that, I'll pass back to Brian for the conclusion.
So what we try to present you is that for the release test, we choose to detect mainly encapsulated viral nucleic acid. And this because this allows us to have a really comprehensive validation package with well-characterized representative spike-in virus, both the NIH and the WHO reference standard, and this within cell bank and harvest matrix. It is true that the LOD that we claim might be underestimated, since a true contaminant will present additional viral nucleic acid that we are not spiking in within our validation, such as transcript or replicative intermediate. But we live with that. It's better to underestimate and know where we are than overestimate. It's for the follow-up investigation that we may use many other type of tool, and this is to conclude on the replicative status of the potential viral contaminant that we identify. So those other tools, we don't need to have representative validation material as we would have need for release test. So, and with this, we would like just to remind that this is a big team effort and that there is a lot of people who are involved within this work. Some in the past, some still reactive. And with this, we thank you for your attention.
Thank you. Welcome, everyone. I'm Martin Machina. I work as a data scientist at Forelli Institute. Closer? Yeah, okay. And I would like to tell you more about our work on developing a novel virus detection pipeline. And so probably I don't need to introduce to this audience the cases where novel viruses were detected in biologics, but I would like to just stress out that there's also other places where we should look for potential viral contamination, and one of them being blood donations, because obviously this poses a threat to the general health of the public, but also to the medical personnel. And so just only in Germany on its own, we see more than 6 million of blood donations per year. And they're being actively screened with a PCR-based method. And also, thanks to this screening, we see only a handful of transmissions over time. But of course, the screening is done only against a handful of selected viruses. And of course, we would like to therefore extend it to all possible known and also unknown viruses that could be potentially contaminating the blood. And so with this motivation, we started to work on a virus surveillance system that would take a small sample from the blood donation, isolate the viruses, sequence their DNA and RNA, and then of course analyze it with our data analysis pipeline to identify known viruses and also potential unknown novel viruses using machine learning. And so currently our pipeline is being implemented in SnakeMake, which makes it for very reproducible execution and also allows it to run locally, so on your laptop or on your desktop, but also make use of the HPC cluster performance. The initial read classification is done with MyCop against the currently reduced RVDB that we do in-house. And then, of course, there is a novel prediction with a custom trained machine learning model. And all these results are then kind of summarized in like a interactive dashboard where all the other personnel can like later on revise these results. And of course, the pipeline is designed to pretty much take any Illumina pattern sequencing reads, irrespective whether they come from a non-targeted approaches, such as those where you take some nuclease that removes the host contamination, or those that are more targeted, such as the VirCap-seq-Vert that kind of enriches for the virus sequences with specific probes. And so this was introduced earlier on by my colleague Gibran earlier in his presentation this morning. And so I would just like to guide you through the process, what we actually do when we receive these reads. So in first, so quite important step, and is we try to remove any non-virus contaminating reads. And so this contamination obviously comes from the host, because it doesn't matter how much you try to enrich for your viruses, there's always going to be some contamination going on. And so there's obviously going to be tons of human sequences, but also, and so obviously also some bacterial ones that come from the puncture wound when the blood was drawn, and also some of the mice and bovine. So what we use is Bowtie2 aligner to kind of align all these sequences against these selected genomes, and anything that aligns is then removed. And then further on, all the reads that pass through are then subjected to trimming and some quality control. So we only end up with the high quality reads that are then further on used for the initial classification into the specific viral sequences, viral species. And so obviously, you probably know that there's many ways how to do this classification. There's many published and also many used algorithms and tools that can work for this classification. But generally, we can probably group them into two main categories. One is being the so-called alignment-based ones. This is, for example, the text maps or MyCop. How they work is they take all the reads and they align them with some kind of aligner backend. For example, this could be BWA, for example, for the MyCop. This, of course, takes some time. And of course, then afterwards, after this alignment, they use some kind of like a probabilistic way how to distinguish if there is some sort of multi-mapping problem. And then there's the other group, which is this K-MER-based ones. For example, Centrifuge or Kraken2. They try to take a little bit of a shortcut to it. They come up with this sort of like a lookup table where they take all the possible K-MER sequences and their associated lowest common ancestor. And so when they try to then classify these reads, they look or they search in this lookup table and they come up with the most probable taxon where this read originated from. But obviously, all these are being reported as very good, and so it's hard to know which one to take for our pipeline and which is going to work best for our case scenario. And so what we did was we did a test for these. We generated an in silico data set where we kind of simulated different sequencing reads coming from different viruses at different concentration. And we mixed them also with potential contaminants, what we assume would be a human and some random sequences. And then we asked these tools, can you find out where these reads originated from, which virus? And so at the basic very low mutation rate at 0.4%, which kind of represents the normal error rate coming from the sequencing machines. They all perform quite well. If you can see from the F1 score, they all almost seem reach the score 1, which is kind of consistent with the literature very well. However, when we try to introduce more variability, more mutations at 5 and 10% mutation rate, which is kind of to simulate the case where kind of this natural occurrence of mutation in the or like variability in the viral population, we see that the K-MER-based ones start to fall short and start to have very reduced performance compared to the alignment ones. And out of all these tools that we tested, we found out that the MyCop performs the best in all this case scenario, which we decided that we're going to implement in our pipeline. And obviously, this is only kind of the half work because now you have the aligner, but obviously you need some kind of right database to work with when you're going to align it against you. And so for that, we chose the reference viral database, the RVDB, which was developed in Arif Askan lab. And so it is very nice because it kind of combines the RefSeq with all the gene bank sequences. So it has quite the breadth of the different variability in the viral population. And so technically we should achieve quite good specificity and sensitivity. However, when we tried to actually test it with our MyCop, what we observed was quite something contradictory. We actually saw that we see less classified reads, and even to the point where sometimes we missed some of the viruses that could be detected. And we kind of try to investigate what's going on there. And so we find out that the data set is actually quite imbalanced. It contains a lot of sequences from a handful of very overstudied viruses. Of course, and these sequences are quite very similar or even identical to each other, or are very, very short, which obviously creates a problem for the myCOP because you get a very many multi-mappers, and the myCOP just doesn't really deal with that very well. And so we came up with this custom filtering set step where we implement this mesh, this min-hash algorithm kind of based comparison, where we take all these sequences in the RVDB and we compare them to each other, how similar they are to each other. And those that are identical or very close in the sequence together, we remove. And of course, we also remove those that are very short, less than 1,000 nucleotides. And this leads to quite a reduction of the database, and also makes it way more balanced in a sense that we don't get some species that have 1 million reads while the other ones have only, let's say, 10 genomic records. So which makes it very well suited for machine learning, but also makes it way better performance for the myCOP classification. And so that's what we kind of did. We implemented this myCOP with this reduced RVDB database for our classification. And just recently, we also added this secondary step of including the Kaiju classifier, which uses this protein RVDB database. So we're using 2 of them together in order to reduce some of the false positive hits. And of course, when we try that into the WHO data set, we can see quite well that we get a very good sensitivity. We are in many cases able to detect the viruses even at 10 copies per milliliter, which is quite good. And those also already covered by Gibran before, so I'm not going to go too much into detail here. And obviously, then what we do with these classified reads, we try to calculate genome coverage from them. And we also try to estimate their abundance of those viral species. And we use those metrics then to further filter some of the false positives from the false negative and so on. So currently, the filter that we're using is required that the positive hit needs to have at least 1,000 nucleotides covered in a maximum of 3 separate genomic regions. So obviously this can change in the future, but this is currently what works for us. And of course, then all this information that we gather throughout the step is then combined into an interactive dashboard report that is then available through browser. So pretty much even untrained personnel can just go through and browse through the results and see what was actually found in that given sample. And of course, sometimes it happens, or quite often happens, that not all the reads can be classified. So there are some reads that just pretty much cannot be classified or cannot be mapped to anything known in the RVDB database. And so in that case, we could assume that these potentially could be coming from a novel virus that hasn't been observed yet and is not in the database. And so we want to investigate whether this is coming from a novel virus. So what we do is we assemble these unclassified reads de novo into longer contigs, and then we try to predict whether these contigs come from a potential viral source using a machine learning model. And so obviously this kind of raises the question, how do we actually train these models, and how do we test their performance given that we don't know if this novel thing even exists, right? And so kind of what do we do generally when we train these models? We take our kind of reduced RVDB database and we chop all the genomes into tiny smaller pieces of nucleotide sequences, and we provide it to a given model. And of course, this sequence is then processed mathematically by the model, or coming through all those different layers of these artificial neurons. And because it's an untrained model, it provides just like an initial estimate what it thinks, if it's a virus or not. And then what we do is we just compare it to our expected results, which in this case should be 100% virus because we know it came from the viral source. And using this information, then these kind of connections between those neurons in these artificial neurons within the network are adjusted. Some become maybe more stronger, some are becoming weaker, and we do it repeatedly until we reach the best performance for a given architecture. But of course, you can imagine that there's many ways how these artificial neurons can be connected, how many layers we can put, how the connections are going to be. So there's many different architectures, machine learning architectures that we could test or try. So for example, there's very common ones called convolutional neural networks, and they're generally used for processing images. So in this case, it's kind of this very hunter where this implements this convolutional neural network, kind of consider the sequence as some kind of like an image where each nucleotide is sort of like a pixel. And so it looks at the context of those neighboring pixels and try to find out where this context kind of matches the virus or not. And then there's, for example, other architectures like transformer ones. They are more used in natural language processing and the large language models. So in this case, this Vertex model architecture, it kind of considers the sequences as some kind of like a language that contains of an alphabet of 4 letters. And so obviously it tries to look whether these sequences, like the sentences that we give it to, are potentially from a viral source. And so we went through the literature and we identified some of the potential potential machine learning architectures that could we test. The criteria were that they had to be published relatively recently. They had to have a publicly available code and need to be at least well maintained in order to work. And so of course you can see that there is a bunch of convolutional neural networks. We have the transformer one. We have the long short-term memory one, which is kind of like, again, used for the natural language processing. And we decided to test which of these would give us the best performance for the novel virus prediction. So how we did that? So we implemented this so-called leave one out strategy. So how it works is that for each of the architecture that I just mentioned, we train it always with some sequences that are coming from the background. So that would be some bacterial and human sequences that we artificially generate. And then, of course, we train it also with sequences that come from this reduced RVDB database. But in this case, before we train the model or provide the sequences to the model, we remove some sequences that come from one particular viral family. And so this sort of viral family then acts as a completely novel one because the model is not aware that the family even exists. And so what did we actually do? We train each architecture 10 times, and each time we remove a different viral family. So we have 10 models which don't know about 10 different families. And then we, of course, test and evaluate these models by providing them contigs, artificial contigs that can, of course, come from the human bacteria and from this viral family that was left out during the training. So now we're asking the model, well, is this sequence that you've never observed and never known about, is this actually a viral source? Is it a virus or not? Okay, and so here is kind of like an example of one of the results that we kind of obtain. For example, for one of this model called Vertex, we collect different parameters such as the recall, which is the sensitivity. So it tells us kind of how many false negatives we have there. And then we have the precision, which kind of tells us about false positives, and the F1 score, which is the combined precision recall statistics. And as you see, just if you consider one of the columns, you are gonna see 10 black dots. So this is the performance of each of those 10 models that were trained for that particular architecture. And the red dot just shows you the median value. And as you can see, for example, for the Vertex, it has pretty good sensitivity across the board for different lengths of the sequences that we try to predict on. However, for example, for the precision, that definitely gets significantly affected, particularly for the longer ones. So we get way more false positives there as the sequences get shorter. Yeah. And so this we can then summarize in this kind of like a little bit busy table, but pretty much the take-home message is that the best performing one was the Vir Hunter, which was with the highest F1 score. It has a pretty good sensitivity across the board of all the different sequence lengths. And of course, it has also quite a good also precision along the way. The next one is Deep6, which has really good specificity, so precision. So I mean, there's not so many false positives, but of course, this is kind of on the expense of sensitivity. So we would lose some of the potential viral contaminations. Then the next one in line is, of course, then the Vertex, Miner, and then Vertifier. And of course, we also tried to go a little bit further. So we repeated this whole test, but this time kind of trying to remove separate classes. So kind of 2 levels up. So we removed all the viral sequences that come from a particular classes of virus and repeated the whole performance test. And so you can see that the performance values are a little bit worse than in case of the family, but the overall order just doesn't change, and it seems like the Hunter is just best performing in our hands. And so with this, we kind of went on and we kind of implemented into also in our pipeline. So we, of course, then we take this unclassified reads, as I told you, we try to assemble them in the context with spades, and then this assembled context, are then given to the Hunter, to our custom trained Hunter model. And these results are then of course, available for this dashboard that are then, of course, able to be reviewed later, whether this is actually a virus or not. Right, and so just give it like a quick conclusion here. We developed this new Bloodware pipeline that kind of is implementing the SnakeMake. So for the reproducible execution, we're using MyCub with the BW backend to kind of classify all the reads initially to the known viruses, and we're using a custom trained machine learning model to then predict whether the unclassified reads are coming from a potentially novel virus source. And we also tested for now the sensitivity of the pipeline, so we're having pretty good limit of the detection of about 10 virus copies per milliliter. Yeah. And so, yeah, with this, I would like to thank to the German Ministry of Health for the funding for this whole project, and obviously all the members that kind of contributed to this whole system. Particularly then there's Marcus, my colleague, that worked kind of hard on all this to implement this pipeline, and of course then Liam and Renate for leading this part of the project. Yeah, and of course, thanks everyone for attention.
Thanks to all the speakers, very nice talks, and for sharing all your details. I had a quick question for Gibran. With the targeted approach, I noticed that you also had human ERF-K in the list that was detected, and I was curious as to how that would be distinguished from cellular sequences, which may also contain that provirus.
Yeah, that was done with the first panel. Now it's updated. And in the new ones, we haven't seen it anymore because it was not intended to catch it. Indeed, that one, because we cannot, as you said, distinguish from something that it's in the human.
Yeah, so a follow-up. So if you're excluding that, is it excluding detection of all endogenous retroviruses or all retroviruses because they share homology? So what is the detection for retroviruses with this strategy?
Yeah, so for endogenous retrovirus, the panel was not intended to detect them. So I cannot give the details because I didn't make the probes myself. So I don't know the details of which ones we can detect and which ones not. But yeah, the intention is not to detect endogenous retrovirus with this.
Marc-Éloi Patoquest. I have a question to Martin. Thank you for your talk. Regarding the contexts that have been detected by this approach, this new tool, how many were also detectable by BLAST?
Yeah, so obviously we need to do more tests, but we had few cases where we can definitely detect that some of those contexts come from the virus, but obviously there's still quite a lot of also false positives, which of course and also requires a little bit more fine tuning on the model or coming for more filtering steps later on. Although this is currently mostly for contigs that are very short, which obviously this is the hardest case. So once we get some samples where we can get larger contigs from the unclassified reads, then we gonna go know more about how it actually performs well. Yeah.
Thank you.
Yeah, I also had a question for Gibran. I was wondering about when you determine if something is detectable or not. You said that 3 of the 5 replicates should be able to detect it. And considering yesterday when we talked about the lowest common denominator, and I was just considering if you wanted to implement this in a clinical trial or GMP, what would you do if the replicate that didn't catch it is the one that you're running on a sample that you are about to release?
Yeah, so this we still we still have to set the thresholds for that. This was just the first try that we made with this panel, and that's why we determined, okay, 3 of 5 will be our LOD, but of course ideally is to detect in the 5 of them. I don't know if you want to.
So usually it's a kind of statistical approach, having replicates and dilution steps, and it's like an endpoint dilution titration with a virus or so you can apply Poisson-based or so statistical mean then to detect a probability of detection. And of course, the less replicates they have and the broader your dilution step is, the more or less accurate you will be with the determination of the detection limit. And I think what we wrote in the pharmacopoeia chapter was this 3 dilution steps in tenfold, at tenfold dilution steps, 3 replicas. That's not much for a PCR. You usually want to have 24 replicas. So it really depends on the accuracy of the detection limit that we want to have. And with the pharmacopoeia suggestions, also with the 3 replicates and tenfold dilution steps, we have to be aware that the LOD that we get out will be quite inaccurate. So the confidence interval will be up to more than 0.5 log or less than 0.5. So we have a confidence interval of 2 log. So we have to be aware it's not the best accurate determination of the LOD. On the other hand, we were discussing it's of course a qualitative test. So it's what you want, and of course a more accurate determination needs more replicates.
And just for the previous question, to contribute a little bit for the marking part, this system was recently established and we tested from some of the Mexican samples. And I can say that by this system, we have found reads that when we blasted, in these contigs that we blasted, some of them were reads that for some reason were discarded by the first part of the pipeline, but belongs to the pegivirus or to the anellovirus. And we catch them with this system, even if the first part was rejected them.
Okay, but they should have been detected by your classical pipeline. So it's a bug of the first pipeline, not an advantage of this. Okay.
Is it okay that I continue? Yeah. I had a question for Song with your internal process control. That you have both a RNA and DNA control, but I was thinking, and maybe also for the room, that the RNA control becomes the DNA control or a similar control after RNA or a reverse transcriptase. Have you considered removing the DNA control?
But the RNA control will control the reverse transcription.
Exactly. And then afterwards, it will
control.
control the same steps as the
Right.
DNA.
So I think that's more like mimic the DNA control is mimic for the DNA viruses, and to capture any potential impact, like from the nucleus, for example, from the the impact might be different for the RNA packaged or the DNA package. So the intention is to have both.
Okay, so it's a risk assessment evaluation to have both instead
Yeah.
of just, yeah, okay.
Maybe if I may, if you work only with your RNA control, let's imagine that you have an issue at the RT step. You don't know if you have some other issue because you will have nothing. But The DNA control allow you to identify only problem with the RT step that doesn't work for your RNA control. But if you have only the RNA control, you don't know where the problem might be. Maybe at the RT step or at another step.
I agree, but if the RT step doesn't work, then we should redo the method.
Yes, but you won't be able to capture some other problem.
That's true.
Also, like after we have accumulated lots of data, we can see the recovery, like the spiking viruses. You have DNA viruses and RNA viruses, and you can see the correlation. Like so during development, we always do with and without benzonides to show that the nuclease treatment will improve the sensitivity, but it doesn't have the equal impact on the DNA viruses and RNA viruses, and actually is correlated with the DNA control and RNA control.
Can I ask a last question for for Martin? And it's actually a little twofold. I was wondering if you have distinct classification of the mutation rates that you are introducing, or if you're just introducing SNPs, or are you? What kind of error model do you have on your in silico generation?
Oh, for yeah, those were just substitution mutations. Yeah, we could technically went also to do like indels or yeah, but I think that would even severely more affect actually the k-mer ones, because then you pretty much have like a shift, so the whole k-mer pretty much is different from the point of the insertion, so that would perform even worse. So like we just went on for just a simple substitution mutation.
Okay, yeah. My next question was regarding the unclassified reads that you make context of. Have you considered retaining some of the classified reads to help in the context creation, or is it just the unclassified that you use there?
So far, we're using only the unclassified. I mean, do you see any advantage of keeping those? Like putting some more.
I was thinking that it could help create the context if the unclassified reads are just maybe bad reads, or the virus differs in some degree from the references, so that it cannot capture those reads.
I mean, there's definitely those cases where we see that there's some sort of like a misclassification of some reads that are thought to be the virus, but actually they are not, and so on. So technically, it would make sense probably later on to do like a pooling of those reads that are just like a misclassified, and then put them back into the pool of the unclassified, and just regenerate rather those contexts using those ones as well. Yeah, I mean, that's it.
And what kind of quality do the contexts have that you create from the unclassified? Pool?
Quality in what sense do you mean?
What is the like N50? How big are they?
So we retain everything that at least have 500 nucleotides, which is like, we don't do anything shorter than that because we also didn't test for this. But they can be as long as they can be, and obviously the longer contigs they are, the better, because then of course that means they're more likely coming from a virus. Right? If you see something that it's only 500 nucleotides and it takes a virus, this could be, as Gibran said, just like pieces of the virus that we have in the pool, but they were kind of wrongly classified or excluded from the classification, right? So if it's actually a novel virus, you would expect something longer to assemble.
And do you have an upper limit? So not be
No,
a bacteria contamination instead, because their genomes are significantly larger than
right. But
viruses.
also some of the viruses can have quite large genomes, so I don't think we applied any cutoff for that, but maybe later if we find out that it's a problem for us, yeah.
I'm done.
Question.
Do you have a comment on this, Arifa?
Well, actually, it was just a quick comment that you indicated that you've taken the sequences from RBDB as being only virus, and you're training your data set on those sequences. As you know, I'm sure, that, we're still working on the annotation of the sequences, and the database contains retroviral sequences, and the endogenous ones contain cellular flanks. So unless you are excluding them, or unless you are, doing something to only use the virus part of them, your training is including some non-viral sequences. And some of the reads that are not aligned could be related to that. So I think was you were saying that the accuracy you had, I think 75-25 or something. And so it might only be because they're not all real virus. I mean, some of them are non-viral. There are some that are vector sequences there, and those have been annotated because we can't remove it. We decided we're not going to change the sequences that we're using from NCBI or from GenBank because we're using them directly. So our strategy is just to annotate the regions that are non-viral, but we haven't worked on the endogenous retrovirus sequences yet on that. We're working on cleaning up the other ones that have non-viral sequences that people deposited or ends and things like that. But just to let you know in the training, just to be aware of that.
Yeah, thank you. I mean, actually, I didn't mention that, but yes, the endogenous retroviruses are removed. So pretty much everything is first screened for the taxonomy origin, and if there's anything that is non-viral is removed. So technically, those are removed, but there probably, as you said, there's probably some stuff that is just not well annotated and still in there, and that definitely might still cause problems. So there might be some more filtering necessary. Yeah. Thank you.
There's a question in the back, I think. Do you still have one? Yeah.
Okay.
Thank you very much. I have as well a question, Martin, concerning the training and the F1 scores that you generated. So one thing that wasn't 100% clear to me, but maybe you mentioned it and just missed it. So the scores that you obtained, were they for the fragments directly or were they for the full pipeline where you simulate then again reads, you mix them with other reads and you go through the full pipeline assembly step and see whether they actually come out at the end?
Right. So this was not yet through the whole pipeline. I think that still needs to be done, but it requires a little bit more testing because, of course, then we need to remove those viruses from the mycop as well and make sure that they're not there. So that still needs to be done. But so far now, this initial testing was done on the reads that were just taken, simulated, and assembled. An extra step. Yeah.
Yeah, that makes sense. I was surprised by the high scoring. Thinking about all the technical steps in between that can go wrong, especially as well in the assembly process. Thank you very much.
Johannes?
Johannes Blume, Paul Elle. Coming back to the nucleic acid treatment, and I remember when we were playing around, we had to somewhat reduce our super potent for nucleic acid cocktail because some virus like the Bunya virus didn't work that well. So a question to you, but maybe also to Bradley Hassan from Merck is, what was your experience in the panel of different virus families with the nucleic acid treatment?
Yeah. The spiking material we have been using the 16 NIH viruses and also the panel of WHO viruses. So far, with and without the nuclease treatment, in our case, benzoate treatment, there's always improvement of the recovery of the viruses, but to different extent. There's no negative impact within that matrix. I think if you have a simple matrix, then more likely the nuclease will impair the viruses. But if you have a matrix with very heavy host background, I think that host background is also sort of protect the viruses. That's my understanding.
Yeah, and I think just along the same lines, I noticed that you said moderate. You highlighted moderate for the nuclease treatment, which may be an important point, because when we did different concentrations of nuclease treatment, we found that the high end, they did affect all of the RNA viruses in the panel, but not the DNA viruses. So maybe some sensitivity, but I think again, you need to test the viruses with the nuclease that you're using.
But it depends on the viruses, and the worst case viruses are not in the panel, like Bunya or others that lack a matrix protein, meaning that if there is a hole in the envelope, the nucleic acid is reachable by the nuclease. So the information we can take from the WHO panel is limited to the list of viruses with a certain structure, which is quite resistant to nuclease, in fact.
So I guess just to finish on that comment. So what we did find that the RNA viruses were sensitive that are in there. So you may want to test those specifically, especially the retrovirus, feline leukemia virus. So there is sensitivity to the RNA viruses specifically. The Bunia, I don't know if it's different degree of sensitivity. We haven't compared that, but but the RNA viruses in the panel are sensitive to at least higher levels. I don't know the degree of different viruses to different concentrations of treatment, but
Yeah, that like Rifa pointed out, we use the moderate nuclease treatment, and in our data set, all data set, there will always a substantial fraction of rays are actually the host rays. I think I checked that is a ballpark number is we usually sequence using P2 flow cell, there will be at least around 100 million rays from host.
Yeah, for us, and I think that's why Johannes made the question. In our case, when we were testing the Bungia and also with the PEGI virus, we found that for plasma that was frozen and thawed, PEGI virus lose a lot of the protection against nucleases, and it was reduced a lot, at least testing by PCR. And for the Bungia, we found that after our treatment, and we don't use so soft treatment, we use benzonase, turbo DNase, RNase A, and RNase T all together. So really to push everything down. And we found a reduction in the Bungia from 15 to 20 CTs by the PCR. Then we reduced a little bit the treatment to don't be so strong. And by PCR was still a reduction of 8 to 10 CTs, but in the sequence we were able to detect it by both the target and the non-target. So probably these enzymes was attacking a specific part that was related with the PCR, and we could detect the rest of the virus.
Obviously, but it depends on the initial titer, no? So if you reduce by 12 CT,
it was like from CT 20 to CT 38, something like that.
Yes, I agree. Might be the titer, but might be also really the background matrix with the host nucleic acid and how it distributes. So I'm really wondering is when it's necessary to revalidate stuff if the matrix changes.
I think it'll be very useful to test the RNA viruses from the panel to see if you see the same effect as you see with the Bunia. Thank you.
Yeah, sorry, I lied, Martin. I have one more question. With the large language model training, you mentioned that it was aware that it was a 4-letter alphabet. Is it also aware of reading frames and codons, or is it only 4 letters?
So it wasn't actually like a large language model. It was just the transformer-based model. Yeah.
Okay, I thought you said it was per.
No, I just said that those type of architectures can be used in no large language model. So
Okay.
as an example, yeah. No, it's not aware of the open reading frames or anything like that.
Peru from US and FDA. So I have a question for Gibran. So I'm wondering, because you are showing the result based on the serum background, so I would like to hear from your comment about would there be any protocol changes if you want to implement this technology to the cell bank testing?
So for example, we have tested also this panel with adenovirus background to 10 to the 9 adenovirus in the background, and works very similar, a little bit reduction in the sensitivity, but works very similar. So because it's not only related to the matrix, but the probes are designed to only capture the viral sequence. Even we don't capture only the viral, we can catch also some human remainings and bacteria and so on. I think in principle, we will try it as it is. If we saw that it doesn't come as we expected, then we can try some modifications, of course.
Yes, because you can imagine that the cell bank background will be much more complex than C serum. So are you expecting any interferences?
In theory, should not, because the probes are specific to catch the viral sequence, and we use the probes in excess to avoid that any competition could read out for something. But as I said, for example, what we presented is the 24-plex, and I didn't mention during the presentation, but when we do the capture after the barcoding, we pull 12 libraries together to do the capture with the 12 libraries together. We try always to have libraries that are homogeneous or to adjust the volume according to the amount of library that we have to avoid this competition. And this can also be optimized. We can reduce the number of samples that goes in one capture pool to increase the sensitivity. We can also reduce the multiplexing to increase the sensitivity. So we have a lot of different strategies and a lot of space to play around to increase the sensitivity in case of any problem with that. But so far is working like that good.
So my second question will be about the design of your probe. So do you take the consideration of the viral genome size for design probe? For example, if you have very long, if you have virus with very long genome, will you have more probe? And asking this because it seems like you have the better sensitivities for the virus with very large genome size, such as EBV and coronaviruses, except for PCV1.
Yes. Yeah, of course, as I mentioned in the probe design is this, each probe has a space without probe between 25 and 50 nucleotides. So if the virus is longer, it has more probes. And it has, in this second version of the panel, tried to be adapted to don't enrich too much the larger virus in comparison with the small ones, because they found, as I said, this was designed by Columbia University. They found in the first version that they were very keen to capture a lot of herpes sequence and so on. They found that and then reduced the number of probes, or the concentration at least, of these probes to try to have a more, let's say, homogeneous panel.
Thank you.
So I have an online question from Trent Bosma at US FDA, and it's for Martin. And he wants to know, how does your reduced RBDB compare to the CRBDB?
Sorry.
How does your reduced RBDB compare to the clustered RBDB?
Clustered RBDB. So the clustered RBDB is already the basis for the filtering step itself. So we don't start with the full RBDB, already start with the clustered one, and we further on filter from that step. Yeah. So.
Maybe just follow up on that database question. I was curious because you showed with the RVDB, the original version, you were not able to detect the RSV. Did you ever go back and see why that was the case? Was it misidentified or something caused it not to show up? Because your filter database is based off of the RVDB. So just curious as to why that is.
I don't remember quite well because it was done by Marcus, by my colleague. I assume it was because there were probably too many similar sequences, and so when there are too many reads that align to multiple sequences, they become sort of all multi-mappers, and then the microbe cannot very well distinguish where in which genome they should be assigned to, because everything is just multi-mapping. It needs some kind of unique mapping in order to decide on the initial probability, and then this initial probability is used to assign those multi-mappers. So if everything is multi-mappers, then it creates a problem. So kind of removing these very similar sequences kind of helps this later, yeah.
So in that case, is that a specificity question where you're then detecting an additional virus on top of what you've put in?
I think if I understood Mike correctly, these are probably then tossed out because it doesn't know what to do with them. Yeah.
Okay, thank you.
So I think here, just to add on that, it fits very well to what we heard. I don't know, I don't remember if yesterday or in the workshop, that the database and the pipeline should match to each other. So that doesn't mean that this reduction that we did for our pipeline will work for others, and for others probably the already compressed RVDB or the big one works better.
Megan, I had a question for you. Your study was really lovely, actually, between the two technologies. For the adaptive sampling, you said you saw human and E. coli for the reads that were like less than 1,000 bases. Do you do any filtering based on size, like less than 200? And is there any merit to just filtering out anything less than 1,000 so that you're only looking at the adaptive sampling reads?
So we have a bioinformatics team that does the pipeline analysis. So I don't want to say I remember every single step of it because I don't. But I do know that we set the sequencer to filter for reads lower than 20 base pairs. When you're setting up the run, you can select 20, 200 and higher. So I set it for 20, and I think our bioinformatics pipeline, I might be wrong here, but I think it filters for reads of 150 base pairs roughly. And in terms of should you filter out reads less than 1,000 base pairs, for AAV specifically, perhaps you may want to, because you want those nice long reads to really cover the genome. But if you have a good quality read that's 500 base pairs, I don't know, you should really throw it out. So I might actually lean towards maybe keeping them anyway.
I actually had a question, if I could, for Megan. I was wondering, when you're sequencing the ITRs, do you have a characterization of sequence variants that might be, and is there like a distribution of population of small sequence variants, and is that something you characterize and have as a quality control, or is everything exactly same sequence and you're trying to maintain that, and can you quantify that?
So the goal of the experiment is to ensure 100% match to the reference sequence to make sure that the AAV sequence is exactly what we expect the patient to get. And one thing that I forgot to mention in my talk is that for the variant construct experiment that I showed, we wish to do limit of detection experiments to really understand the limit of detection between the platforms and between the GOI and ITR to understand the differences that we see, and which ends up what ends up being the limit of detection. So we still need to find out.
Hi, yeah, Charlie Swafford, MIT. Megan, this is for you as well. It's a great talk. To be honest, I was a little surprised that the adaptive sampling worked as well as it did, just because of the size range that you're looking at. I mean, at least in our experience, adaptive sampling for an adventitious agent detection has not worked that well from an enrichment standpoint, just because the reads are usually too small, even though it's long read sequencing, for the GPU to make a decision whether to reject a read or not. So by the time it decides this is a viral read or this is a human read or whatever, it's already passed through the pore. But if I remember correctly, and admittedly it's been a while since I did it, I think you can collect some statistics on the adaptive sampling in terms of how many reads were rejected or accepted and so forth. So I don't know, did you collect any data on that? And do you have a sense of when you did adaptive sampling, how many reads were rejected or accepted, and what those, and I don't know, like, do you know what those reads were?
I'm getting a very short answer. No, we didn't check the rejection statistics. I'm sure we have them, so I could take a look, but no, I don't know off the top of my head what percent of reads were rejected. And to your point about adventitious agent detection versus AAV adaptive sampling, because AAV we hope is very uniform and long read, so the adaptive sampling can kick in and not let those shorter reads pass through. So I think maybe that might be a difference between adventitious agent detection and AAV sequencing, or maybe we've also tried depleting against host background for adventitious agent detection. So we might also want to consider trying that for to deplete like CHO, for example.
Okay, thank you. I mean, maybe one more question, not just for you, but maybe for the audience, because this is more an adventitious agent question, but you did a nice comparison between short read and long read sequencing. And I always kind of wondered, at what point is the quality good enough for Illumina versus like maybe you're getting Q30 versus Q38 or whatever, but does that really matter at a certain point from an adventitious agent detection perspective versus the fact that you're getting a read that's, you know, way longer? So I guess where's the trade-off here in terms of quality of a read versus the length of a read? And at what point should we start caring about one versus the other? I'll just leave that at that.
So I'll just share my thoughts about that. It's a good question, and you know, I think there's been thinking about that. So to me, I view it as detection versus characterization. You know, for adventitious virus, you know, it's detection. So you do want accurate detection, whereas if it's characterization, our approach is, you know, you want to get the long reads, but then we combine the data, you know, for more accuracy with the Illumina data at this time. But that's where you want to get the full virus genome sequence. So
Yeah, when you want to detect sequence variants and you want a very good LOD for detecting sequence variants, perhaps the better quality is better.
My question is also to Megan. It has been reported that nanopore sequencing might have problems with structures because as the DNA passes through the channel, then it forms the structure again on the other side, which will have an influence on the speed of the rest of the DNA coming through. And I wonder if it is a problem. I mean, this could be reflected in by your coverage plots, where the ITRs have higher coverage than the rest of the stuff. Have you considered separating the two things? So, you know, concentrate for the flip-flop orientations by sequencing the full length but cut away the inverted repeats, and then you go for the gene of interest, the separate sequencing, or is this not an issue for you?
I hadn't heard of the secondary structure affecting the speed of translocation through the pore, so that's good to know. We haven't really noticed anything on our end. Yes, you might have seen in the coverage maps, the best coverage was actually at the ITRs, and it did get lower towards the middle. We figure that's because of the ligation step. It's ligating the adapters to the end of the AAV sequence, and so maybe reads are shorn during the process of nucleic acid extraction and library prep, and so you're not getting the full length read in some of those reads, and so they cut halfway or a third of the way through, and that's why we get that dip in coverage. But that's our main explanation, but we haven't really tried to specifically sequence the ITRs or specifically sequence the GOI in separate bins.
Yeah, Megan, I said that we would talk later, but there's obviously an interest in adaptive sampling, so I'll just fire away now. I was curious, I have a couple of questions regarding it, but are you using the onboard algorithm for the adaptive sampling?
Yes.
Yeah, okay. And what speed do you sequence for it?
We sequence at super high accuracy base calling.
420 or something?
I think so, yeah.
Yeah. Have you tried with the fast 240, I think?
No.
Okay. Because they say that they can make the decision after 250 bases. So if you are firing through with 400, then yeah, that could be an issue. And how do you? It can run without a bed file for hashing, but do you run with a bed file? And if so, how do you configure it?
No, we're not running with a bed file. I think we're running with a FASTA file, I believe.
As the reference?
Yeah, as the reference for adaptive sampling.
Okay. We have experienced that if you give it a bed file that chops up the genome, it can take the decisions faster because hashing is very, very fast. Okay. That was it.
I have a question from Trent Bosma, and it's for Megan. And he wants to know if you had any issues with duplex reads with your nanopore data.
Duplex reads, I'm not the issues with duplex reads. I don't think so.
Back here then. Megan has the mic, so we can stay with her. So this is Devin from Gene Data. Since you have this nice long read data, have you noticed any differences in truncation detection on your AAVs when you're doing adaptive sampling versus not? Does that have an effect in the truncations you see or the hotspots?
We haven't yet looked at truncation hotspots, so I can't really comment on that yet. That's on our to-do list for seeing what our nanopore data can give us.
Cool, thanks.
Maybe I can ask a question. I would like to come back on the validation as it has been performed by Sanofi. And you mentioned that you validate using a 16 virus panel. And I would like to know if you identify some differences depending on the families of viruses. And secondly, did you find some matrix that would be less sensitive than other one?
Yes, there's definitely a difference for different matrices. Like we have heard in other people's talk that there's a matrix effect on the sensitivity. And in terms of viral families, so my experience is, so the very first matrix that we have this issue, like, need to reduce the haystack to find the needle. We found that after we reduce the haystack with the nuclease of benzonase, the improvement for the RNA viruses is a lot greater than DNA viruses. That's we see. And my interpretation is somehow the host background, whole nucleic acids might interfere with the converting the RNA to cDNA. So that also apply almost to all the RNA viruses within that 16 virus panel. So it's like a 10-fold more improvement if you look at RNA viruses compared to DNA viruses. Still for DNA viruses, they are still at least 10-fold increase, but for RNA viruses, the 100-fold increase if you look at the with and without benzonase. So it's really like, it's matrix specific. So for some matrices, it's definitely better.
And maybe that's the reason why we have to assess the metrics to see if it's different or not, and to reassess if we are reaching the level of detection that we wanted to have, or if we need to redevelop and to optimize some steps. The beauty of the reference material.
Yeah, it would be useful. Yeah.
Hey, this is Vanessa Sarathi from MSD. My question is for Martin. I was wondering what is a different or a separate genomic region? So like what are you considering that since that's like part of your criteria, right? Is like a coverage of 1,000, right? And then 3 separate regions. What is that exactly?
So you're talking about the filtering step, yeah.
Yeah.
So pretty much when the reads align to the particular virus genome, they sometimes don't have a full coverage. Let's say you don't get a full genome coverage, but sometimes the alignment could be spotty because maybe some of the sequences of the virus got lost and so on. So we kind of look at those different spots. So could it be that you get some good coverage at certain spots, and we kind of combine those spots together in order to get, so there's a maximum 3 spots, yeah, in total of 1,000 nucleotides, yeah.
Yeah, just to follow up on that, what's the rationale behind having a maximum of 3 spots or 3 contexts?
I mean, all these filters are just very just anecdotal, let's say, by experience, what we see in the data and when we evaluate the results and what we see where the sort of trying to find where the correct threshold is for what should be considered as a real or not. So this kind of very empiric, but yeah. So.
Yeah, one of the ideas behind also was that in PCR, when you do a PCR for a specific region and you want to be sure that you really have the virus there, and it's not just this small spot, one of the criteria by PCR is that you have at least 3 different regions of the genome. That's why we put the number 3 on there.
And
it is not the maximum, it's the minimum that we need to find.
Yeah, that changes things. And of course, I just assume that you need perfect alignment, but is that the case, or is it, do you allow for differences between the context that you have and the reference that you use?
We allow difference, but the percentage and so on, that's better Martin to answer.
I'm sorry, can you repeat that?
If the when you have the context and you align them to the reference, do you have a lower limit for alignment percentage or score or whatever you want to call it?
Now you're talking about the reads or
the context that you create or the consensus of the context. I don't know if you distinguish between reads and the context.
No, I mean what we when we classify and we use the reads, right? So there is definitely some criteria that is inbuilt into the MyCop. So there is some amount of quality and also amount of coverage, which I don't currently recall exactly what the thresholds are, but yeah, we could talk later. You could ask my colleague Marcus who kind of define these criteria.
You can answer now.
Hello. Yeah, we don't use contigs, but we use the single reads for alignment, and we just do standard BWA alignment. So everything what aligns, and everything what is unclassified then goes to Martin's artificial intelligence pipeline. But for BWA, there is not a certain threshold.
Actually, I had another question, general one. Just when looking at alignments and trying to identify viruses, and you have, want 3 spots, say, on a sequence, are you looking at beginning, middle, end? Can it be like anywhere? Do you sometimes have, for identifying a hit, should it be somewhere in like, one hit in the first 30%, middle, and last 30%, or?
Maybe Marcus can answer again if he gets the microphone.
Yeah, so what we do is we align our short reads against the genome, and then we create a bed file and fuse those regions. And overlapping regions will be fused, and we don't have criteria where exactly in the genomes the regions need to be. But our quality criteria is we need at least 3 non-adjacent regions in the genome. And then, the filter is passed. So alternatively, if we don't have, for example, we have full coverage of the whole genome, then it's obviously only 1 region, but we have more than 1,000 base pairs covered. So that would also pass the filtering criteria.
Thanks. I'm just taking this directly. So I'm hearing a lot about mapping and the problematics of multi-mappers and the fact that we have this growing databases with a lot of redundant sequences, and then we are trying to collapse this again with either hashing or other aspects. So I'm wondering, did anybody already so far try to dabble into, genome crafts or variant crafts in order to reduce that to an extent so that we really capture the full variants that we actually can observe still, and reduce as well the multi-mapping aspect, because all those things that are shared between multiple species from the same family might be then actually redundant and help us better in this kind of decisions.
So maybe I'm not the right person to answer this question. Maybe it's better to ask this question to the RVDB people. But you're right that the multi-mappers, they create the main problems. And for our pipeline, we could think to assemble the reads. But we decided against it because we have blood plasma pools of sometimes several thousand people. And we don't want to assemble chimera. So for our purpose, it's better to use the single unassembled reads, but then, I mean, it's normal with extensive databases where you have high variation and many closely related entries, then you will have multi-mappers. I mean, you have this for all the different classifiers. Also, if you do BLAST, if you BLAST your short sequence, then you will also have multi-mapper. You also have to decide where to assign it. Also for K-map-based methods, it also cannot assign it to one specific entry, then it will go like in Kraken 2 to the lowest common ancestor. And that's actually one strength of MyCop, because it tries to still assign those multi-mapping reads. And it does that by not only looking at the single reads, but also at the entire read data set. So it looks at the percentage of uniquely mapping reads. And then in a probabilistic way, it tries to assign these multi-mapping reads to a certain entry. But you need some uniquely mapping reads, otherwise it won't work.
I just wanted to mention about the RVDB. So I think people need to understand that the purpose of the unclustered RVDB was to retain the diversity of sequences. And that also helps in terms of detecting distantly related sequences. And the clustered RVDB was to reduce the redundancy of 98% sequences. So it is serving the purpose it was meant for, but it also allows the users to download it and then sort of, customize it based upon their questions and their pipelines. So people, so there is a reason for the diversity that we want to retain that.
Yeah, I mean, absolutely. And so we kind of get used to took use of that, and of course made our own filtering based on our needs. Yeah.
Yeah, I just want to make an additional comment. Please join AVDTWG Subgroup C. Because, I mean, seriously, we have this kind of discussion perhaps in these 4 years during pandemic. Everybody are struggling, not only us, okay? I think NCBI staff is also joining right now, online, remotely. They also realize there's a problem, so they actually they delay one gene bank release because of SARS-CoV-2, because they really just like us, they also struggling it. And just like Rifa say, NCBI or us, we cannot just simply have some sort of the criteria and decide what SARS-CoV-2 sequence we want to keep and what we don't want to keep. So it's all we want to keep the completeness of the database. So it all depends on your internal pipeline and also your risk management to decide if you want to reduce the SARS-CoV-2 redundancy. But again, I think to me, things like we have, because, the subgroup C meeting is a hard meeting, and we haven't had a meeting for a while. So I think that's a really good chance for we to restart the discussion about so many topics. Yes.
And I just wanted to. Oh, it is. Hello. Hello. Yeah. I just wanted to share some thoughts on this because I've been talking a lot with an ancient DNA guy, which is a totally different field, but some of the issues that he faces with very, very fragmented and poor DNA quality could be some qualities that as an advantageous virus detection method could also use in finding very, very low amount of viruses. And one of the things that they have done is to create a family reference database where you collect, of course, families and then have them represented as a core genome, which could also be applied to maybe viral families, and then have the diversity in the family as paths to and from this core genome. And I have been wondering about implementing a strategy like this, but it's not going to be in our original method, that's for sure. But just to plant the seed and maybe get some input or thoughts about it down the road, because I think it's a very interesting approach.
Yeah, so I guess without knowing more details, what you've described is already exists with NCBI. I think to me it sounds like the RefSeq and the nearest neighbors.
Are you saying craft or graph?
Graph. Sorry.
Graph. Yeah, it's a
Not
graph
graph.
genome. Yeah, exactly.
Yeah. Sorry. Yeah. Yeah.
Yeah, just like that. Mm-hmm. Just have that as a reference.
The pangenome graph approach is still an unsolved problems, and one of the issue is to storing the index of the pangenome. That is really huge.
So definitely, please, follow up on Peru's suggestion to join subgroup C, because clearly if it's something that needs to be considered or thought through, it will be nice to have presentations on that.
No, yeah.
Nobody else online? Nothing? Okay, if that's it, we can break a little early for lunch.
Discussion.
Okay, yes.